Hey everybody, welcome back. Today we're going to start with microbiology, and the first topic is bacteria. So the morphology can vary. The cocci are going to be made out of circular structure, and the coccus is one, diplocus is going to be two. The more common ones that we need to be paying attention to are the streptococcus, they're beads on a line, and the staphylococcus, which is like a bunch of grapes. The bacilli are going to be rod-shaped structures, and the coccobacillus is going to be like a combination between a coccus and a bacilli. The bacillus is one rod, diplo is two, strepto is once again rods on a chain, and the palisades are kind of like zigzags. So these ends will join, that end will join. This is an unnatural one, but those are the palisades. The other ones, more importantly, like Vibrio, is like a comma shape. The Spirochete is a circular corkscrew shape. And the true corkscrew is more of a parasite. So this is Borrelia burgdorferi, is a bacteria that infects ticks. So this is the one that causes Lyme disease. So most bacteria are monomorphic, so they assume one of these structures. There's only a few that can change its structure. Okay, so what do we need to know? All of them have a cell wall and a plasma membrane, the cytoplasm, ribosomes, and the nucleoid, which is basically an open nucleus. It contains their genetic material. Some of them have plasmids, which are the extra genetic material. Is not part of the actual genome. Pili, flagella, some of them form spores. Yeah, so not all of the bacteria have all these structures though, but all of them have these. These are like the basic structures. Cell wall will carry the virulence factors, and this is where the antibiotics work along with our immune systems. It's useful because of gram staining and acid fast, usually made of peptidoglycan or murin, which are units of N-acetylmuramic acid and N-acetylglucosamine. The bacteria will have the N-acetylglucosamine, which is NAM, and the D sugars on the outside. So a lysozyme will come, recognize it, and break that glycosyl bond, breaking down the membrane. Okay, so bactoprenol transport monomers across the cell membrane, and the transglycosylase will make the glycosidic bonds, and those will join the sugars, like right here, right here, right here. Carboxypeptidase will remove the D-alanine to release the energy in order to cross-link the peptide chains. And the transpeptidase will make the cross-linkage. Okay, so a gram-positive bacteria is going to stain with the crystal violet stain because it has a thick cell wall. So the cell wall is made out of teichoic acid, lipoteichoic acid, and pentaglycine bridge, and all this makes up the peptidoglycan. And the crystal violet will stain this and isn't washed away. Okay, so the gram-negative layer has two layers. The outer phospholipid bilayer, it has an O polysaccharide on the outside, and in the inside, the core is going to be polysaccharide lipid A. And the O antigen or the polysaccharide is going to determine the serotype, and the inner is going to be the endotoxin. So the O antigen is what our cells of the immune system recognizes. The transport protein is going to lie within the cell wall, and there's a thin layer of peptidoglycan separating the two cell walls. So the peptidoglycan is actually where the periplasmic space is, and that is going to contain enzymes such as the nutrient transport enzymes. When we stain it with the gram stain first with the crystal violet, that stains the outside, but when it is when it's counterstained, that will break down the outer membrane and stain the inner peptidoglycan. So gram negative is going to appear pink, gram positive is going to appear purple because of the crystal violet. I think the stain for the pink is hemosiderin. That's not too important, but useful to know, I guess. Okay, so gram staining is pink and purple, acid fast, there's blue. So why is the acid fast staining useful? It will bind to the glycocalyx or the slime layer, which are loose polysaccharide protein layers. They are useful to the bacteria, not great for us because they are poorly antigenic and anti-phagocytic. So they don't present very well. So our cells aren't able to recognize it and phagocytose it. So it is very virulent. The membrane is a lipid based, and they have polymers woven within it, made out of mycolic acid again, but this time we add trehalose dimycolate and lipoarabinomannan. A biofilm is going to be a polysaccharide that surrounds like a bunch of cells, and that like helps stick them together and protect against phagocytosis, dehydration, and antibiotics. So these can adhere to the artificial devices. So we need to keep in mind the prosthesis the replacement, like so joint replacements. Like we need to keep those in mind. Any rods that are going to be inserted into the body. So the mycoplasma bacteria do not have any peptidoglycan, so instead they have like a peptidoglycan and arabinogalactan polymer, and that is surrounded by the mycolic acid, and that kind of forms the capsule. So they don't have a peptidoglycan like cell membrane itself. Capsule can have a variety of shapes. There are going to be the sterols in the phospholipid bilayer, and they're going to be bound together with a phosphodiester bond. These do not stain with the normal staining methods. So the capsules are very useful to the bacteria because they are resistant to the antibiotics that target the cell wall. They protect against phagocytosis and dehydration. Okay, so an L form is going to be made by the lysozyme, and a cell wall synthesis inhibitor. Antibiotics are going to act on it, such as penicillin. Protoplasts come from a gram positive, and that makes a stable L form. And spheroplast from gram negative bacteria retains the outer membrane. So those make the unstable L form, which can revert back to the original form if it's moved to like a safe environment for the bacteria. There aren't any sterols in the cytoplasmic membrane, but it's pretty similar to the eukaryotic cell membrane. There's the phospholipid bilayer, the semi-permeability, and they have a polymerizing enzyme to make the cell wall. Okay, so their nucleoid is one single circular double-stranded DNA. There are no introns in it, and it's haploid. They have an extra chromosomal genetic material called a plasmid, and their ribosomes are going to be size 70S with 3S and 30S and 50S subunits. Okay, so some have a flagella made out of the flagellin protein, and those will have variation in the amino acid sequence, which is called the antigenic variation. So these are capable of being regenerated if there's any problems. Number and the position vary. There can be a hook, a basal body, and the filament. So the tip or the hook, here's the filament, and here is the hook. And the axial sheath is where, or axial filament is where the flagellin is going to be covered by the outer sheath. The pili, or the fimbriae, so it's like this fuzz around it, is made of fibrin, adhesin, and pilin proteins. The adhesin can go through the antigenic variation to avoid the immune response or to bind to different host cells. Mostly these are on the gram-negative bacteria, and they mediate the attachment to the host cell and transport of the bacterial material. And these are capable of inducing a specific immune response. So maybe they land on here, which is like what, CD40. CD40 might like call in another cell, bind to CD40 ligand, and then a neutrophilic or macrophilic response will occur. Spores are going to protect the cells from environmental stressors, and inside the bacteria is going to be senescent. So it contains only like what the bare necessities are, and they can survive up to years. The spore coat has a high concentration of calcium bound to the dipicolinic acid and keratin-like protein. Sometimes there's such thing as an exosporium. So the core contains DNA, then it's inside of the cytoplasmic membrane. Then there's the spore wall, and there's a cortex surrounding the spore wall, separating it from the keratin spore coat. So it is pretty well protected. What bacteria use it? The gram-positive bacteria, which are the aerobic bacilli, of course, because it's in the environment with oxygen. Oxygen is generally toxic, along with anaerobic Clostridia. Gram-negative is going to be the Coxiella burnetii, and it's also in the tetanus virus, which is why, or bacteria, which is why when there is a cut in the skin, tetanus is able to come in, infect, and more importantly, the spore allows the tetanus to live in the ground. The only way for like true sterilization, so this is killing like complete decimation of every single thing on whatever tool you need, right, or surface. So this is killing the virus, this is killing the spores, everything. Excuse me. There are only a couple instruments / sporicidals that we can use. One of them is an autoclave, and it's basically steam under pressure. Very, very hot steam and very, very high pressures. Dry heat sterilizers exist. They can be like flash sterilizers. They can be like just the common types of autoclaves. But the sporicides we need to know about are the performic acid, vaporized hydrogen peroxide, a version called Paradox, and Oxyvir. So these are very good to not just disinfect. These are good to sterilize the environment, but they're toxic when encountered with the skin. That's it for now. Thank you for your attention, and good luck studying.